SmartLab lecture series, and I'm presenting the key concepts of what we are planning to do. And be aware, this is all under development, and I'll just try to outline where we are, what we still have to do, and what the benefits of this whole developments are. So let's just start. And since we are a small group now, please, whenever you think, raise your hand if it's possible. I don't know if it's possible. Or
it is possible.
Tim,
there's a button on the left, center
but
left.
I don't see the stuff because of my presentation screen.
Okay.
But please interrupt me. Then just switch on your microphone, interrupt me, and ask, because if things are unclear or it should be more detailed, then I'll be happy to answer, because we have a small group here. Okay. So most of half of you, let's say, know already the concept, and I hope that, I mean, these like Kata or Stefan, Tim also knows it. They might be a little bit bored. I don't have substantially new ideas here, but I hope that since it's also recorded and here a few others that are not. That don't know the concept before, still have some nice information to talk about. So let's see. What is the vision? The vision of this whole software idea that I'm developing together with a lot of people. So it's a constantly growing community, and I'm very glad that it's growing. And I hope that with this talk here, can also inspire some more people. What we are trying to achieve is something very simple in its idea. So we all, as scientists, want to have our experiments, our data in a single place, and we want to have it reproducible, and we want to understand it deeply. Maybe we want to also send it to some AI or machine learning algorithm that it can learn with it in a good manner. So in a way that the machine can understand the data. It's not like an Excel file or something which is completely uninformative to a machine, but with a deep information, deep knowledge of the data and of the whole experimentation. And but as some of you already know, that data production and data archiving, storage is quite tedious. So especially if you want to really annotate it semantically, if you want to edit with metadata, with ontology-based terminology, then it gets really hard, because you don't want to do this by hand. So you need like tools, and you need like automated pipelines that do that. It's too much for a scientist. Scientists, they just want to stand in the lab and do the research. They want to draft what they want to execute, and that's it. Maybe do some calculations, but not too much of a data structuring approach. And then for the people that start to think about automation and execution of workflows by a machine, they want an automation execution environment. They want an orchestration of the whole devices. They want some workflow that they can run. And finally, the people that think a little bit further and deeper into the data, they want AI machine learning ready data. So that's basically the common ground, and this is where we try to provide tools and a tool set, a tool chain, a whole suite of modules that help to support this idea. Okay, so very quickly, this system should be able to plan the experiments, to execute the workflows, to capture the data, to query the data, to evaluate the data, to share the data, and to feedback and refine. So very simple in its concept, but very difficult in its design. And here's what we came up with over a long time of experimentation and thinking. So this is what you see here is the third generation of this type of research data management software architecture I'm developing. So I started with a C++ in development. Then I went to R, and I got quite far with this R approach. But then I realized that the language R is very, very slow. It's a very powerful language on the one side for statistics, for visualization. It's quite nice, but it's very slow in processing. Sorry? Oh. But it's there's some microphone on, Tim.
Yeah, it was Christoph. It's fine.
Okay. So R is very slow. So I decided to go for a fast environment. That's why I decided to use Python. Python is also very universal. So most scientists speak or learn Python from the very beginning. I mean, it's coming. So our students in Greifswald here. So I'm from the University of Greifswald, by the way. They start to learn Python. They learn Python courses. So it's a good language. It's a universal language, quite capable of doing very simple and very complex things. And so I use that. And I was starting to a long time ago, actually. So I'm responsible for a large protein screening platform, so as a biochemist. And we had this complex platform that, or still have it, and I'm running it, that produces a lot of data and needs to be structured, processes, execution. And this environment that was provided by the vendor was very limited, so it could only execute things with proprietary drivers, and was not open, the system. So I decided to develop my own system first, and then I realized that it should be more general, more generic, because it's something that many people need, many people want to have, and that's why I also try to advertise the concept a little bit in these groups, to find more people that have this need and to develop such a system together. And what we need, of course, is modules that help to plan experiments, processes, materials, substances, to store them, to make, first of all the components of an experiment available to the system. So knowledge about the devices that are used, the parts, the substances, the organisms. So all of that, all the procedures and processes should be stored in one place, or assembled in one place and made available to the system. And then, for example, the processes, they should be then executed. So they should be possible, or it should be possible to execute them directly out of the system. So you should say, now trigger this process, or schedule it for processing, so long time scheduling. And then it should be sent to a separate service, so independent of this database or this structuring part here. And it should be orchestrated and scheduled. And luckily, we got some money, Katharina was also involved in this. It was a Kiwi project, and we could hire Stefan Mark, who's also here in the audience, and he developed then this orchestration and scheduling part here, this service. Together here at so we used our platform as a kind of very complex scheduling environment for that, and so he could learn a lot about intricacies, complications, and complexity of scheduling. And we decided to, since I was invited to join the SILA working group, we decided to Silanify, if you want, so convert every device in our lab, and there are quite a lot, into SILA devices. So SILA stands for standardization and lab automation, and is an open standard for communication to devices. And we don't use it only to control devices, so it's not like a driver's system, but it's also used for machine learning, communication with machine learning, or even with humans. So we have some very simple implementations. So Stefan made the first ones, and currently develop more complex implementations that we have, like, can send all this scheduling also to human people, so it's not limited to robotic system, automation system. That's very important to understand. What we want to achieve is a kind of system that stores the process and executes the process, or like give the scientists some workflow in hand, but then it should also get feedback from the executions, the executor. So it could be a device that tells, okay, now data is produced, or human that gives feedback, something was added, or something was changed, or something was somehow something happened. So and all of this information is then collected via a separate instance, a separate microservice that communicates with an orchestrator. This is something that is currently under development, so we are exploring this inbuilt communication infrastructure that LARA provides. So the SILA communication infrastructure and the RabbitMQ infrastructure to get notifications from different agents that something happened, an event, and that triggers then, for example, data collection. And the data, and that's very important, should be stored in an open and accessible format, in a linked format. And therefore, we decided mainly to use JSON LD. This is very, very convenient, because JSON by itself is easy storable in, there's a lot of good support for databases, like PostgreSQL database, which we use for the data storage. So the tabular data storage. And the linked data part, LD stands for linked data, that helps to reference to external ontologies, which was partly developed or reused. Depends on what we do, or how good these ontologies are. So we can link then the semantic information to the tabular measured data of, for example, devices like a plate reader or a global thermometer. And this linked data information, so the semantic metadata information, if you want, that is not stored in the tabular or relational database or PostgreSQL database, but in a triple store. So, and we are using the Virtuoso triple store for storing that. The reason why we choose to have like a dual representation of everything is, well, it's not really duplicated. It has different purposes. So the data that is tabular, like for example all the chemicals, like imagine you have like, if you do like a screening process, you have 10,000 of different chemicals, even more, especially if, what we also do, we do like virtual docking. Then you want to dock like several thousands of chemicals against, for example, protein. So protein data, that is very good to be stored in tabular relational databases. But if you want to store like hierarchical or network structure, then graph databases are much more powerful, and therefore we store all this semantic information in a triple store RDF based. So there's an international standard resource description framework, and this defines how you store linked data or semantic data. And the good thing about, or the nice thing about these triple stores are that they come usually with SparkQL endpoints, which is SparkQL is a language for querying these complex triples, and these triples, these semantic information can then be quite complex queried, and especially federated queries are possible for that. So you can combine different data sources like Wikidata, PubChem, Uniprot. There are quite a lot of SparkQL endpoints around there in the world, and there's a unified standard for querying them, and you can combine information from many sources to what you are looking for. And the last component that we also need is a synchronization module, and this is currently developed by a company which is also financed by this Kiwi Biolab project, and they develop like a synchronization microservice that helps to exchange the data. So that's the this part, and so you can basically build up a whole network of these LARA instances in different institutions, universities, or other companies, or whatever you like. I mean, it's open source. There's a Docker container that you can, or let's say a Docker compose file that you can just execute if you have a Docker environment, and then it's very simple to install such a thing, such a LARA instance. We are planning, and this is still under development, to connect also to larger data repositories like Dataverse or Zenodo or the European Science Cloud, with data that is somehow curated and refined and finalized for publications. Of course, what we don't want is to spam the data, to spam the world with raw data that is not useful. So what we still need to envision is a filtering system or quality management system that helps to decide what data should be really kept, what is relevant data, and what is spam. So this is something we still need to figure out. First of all, we provide the infrastructure to produce data. So, and here are a few ideas, a few visions on how this looks like. So, so this is now a real view on the current user interface of the front end interface of this database part of LARA. So it's not the full thing, of course. It's just what you can use for the database, and you have like these sections here, like processes, sequences, reactions, substances, structures, material parts, devices, labware, samples, organisms, data. And it's a modular system. So if you look at the GitLab repository, you see that each of these, you see here as a kind of bar, is represented as a module. I'm using Django. That's why all these modules are called Django somehow. Django is a Python library for making these nice interfaces here, these web interfaces, but it's also an object relational mapper on our M system for making database access quite easy. And the current power and strength of LARA is not this user interface. I mean, we are planning to build this to a kind of electronic notebook system, next generation electronic notebook system. What the strength currently is this very extensive API. And we are using gRPC, and each single table in this database, there are lots. So if you go to the admin view, you see all the tables, and you see it's a lot. It's really a lot. And each of these tables consists of a lot of columns. So for example, just that you get an impression, let's just go for the substances, for example, just because this is something that many people understand. Just select one, ethanol here. You see substances, information about substances. There's a lot of information stored, like the CAS number, the IUPAC number, the PubChem numbers, so all these kind of identifiers. Then the masses, of course, in different versions, like the exact mass, the monoisotopic mass, the molar mass, the density, the physical state of matter. And if there, I selected these fields that are very, very important for chemists. I'm a chemist background, so I can decide what is important. But there are many more properties, and so I have a JSON field here. If you have like much more properties which we don't cover in this rigid database scheme, then you can add much more of these information in JSON fields here, also for computed properties. Some formula, for example, the smile string, iso smile string, the can smile string, in keys. So a lot of these IDs, a lot of these 3D, 2D representations, codes can be stored, logp, xlogp information, elogp, melting points, boiling points. So so you see in each of these tables, what I showed here to you consists of quite some columns. So it's a lot of information, and everything is linked together. So so it's referencing. So for example, if you have like an experiment, then the experiments, they reference, for example, to other experiments. They reference to processes, to procedures, to substances, to polymers, to mixtures. So what you use in the experiment, the devices, the labware, the parameters, the samples, and all of that. All these relations you don't want to add manually. So I'll try now in the next steps to explain a little bit how these combinations are made. By the way, we are also planning, developing right now a Zotero-based literature module, which is also interesting. So how do we get the data into the system? First of all, the data needs to be somehow produced. And for that, we currently developing a language, which we call Python Lab. This language is close to English, but it's still Python syntax. But it's not executed as a Python, directly executed as a Python program. So what you see here is a kind of symbolic or abstract representation of a workflow. And it's easy for even a non-programmer to read. So you have here this robot arm move a container from a source location to a target location. You have an incubation step with a certain duration and temperature. You have another move step from a source location to target location. And then you have like a single read step in a spectrometer at 660 nanometers at 305 Kelvin, and then you have another move step. So things are quite easy to read for humans and for machines, and that's the beauty of this whole system. We do then in the background quite some magic to convert this to something executable, and then we execute such workflows with for example, the orchestrator of Stefan Mark. So we formulate quite complex process like this one in a few lines of code, and then we can convert this to, or map these to devices in our system. And this is quite generic. So you can, if you have your own device, you can just put them in and schedule them and orchestrate them. And for the orchestration, we are using this LARA protocol, or communication protocol standard, which I just mentioned, which is very easy to implement and helps us to convert even devices that has not been designed for LARA before, because you can somehow wrap your LARA device, your device into a LARA device. So you can build like an interface to that, an abstraction. Okay. And then we store this data that is coming from these processes. So here each process with a lot of steps produce some data, and we try to store as much information as possible. So the organism information, the material information, the physics, the substances, the reaction mechanisms, process information in one, as I said, in this LARA database. I showed you a little bit the interface. And then still the question remains, how does the data comes into this database? And the first step is that we have to convert the data usually from a proprietary data format, like, for example, CSV format or XML format of a vendor, or even worse, of binary format, and then make it like available, enrich it with some semantic information. And for that, we develop here on this link a software module that reads this proprietary data and converts it into JSON LD. And the only thing that some developer needs to do only once, is like a parsing module needs to be written for each single type of data format. So because, I mean, the system can't guess what this data looks like, the structure. So some programmer needs to write once a module for a certain software output, and then it can be run easily on all the data that you produce. So, and we are right now, and I really would be happy to get some more example files from people that we build up a nice library that it's much more convenient to use this lab data reader for the next generation of scientists, because once there's like a basic set of these parsing files, then not every scientist need to regenerate these or reinvent these parsing things again. And the nice thing about this lab data reader is that it already enriches this CSV files with semantic information, because the parsing process guides and adds semantic information. So while you're parsing and reading the file, you already decide of what type this is, so what type of measurement it was, which device it was measured from, when it was measured. So all of this data, that metadata that already is available, is converted into these triples, or this RDF representation, or linked data, first linked data representation in JSON LD, and later then into triples in the triple store. So we try to build up the semantic knowledge graph from ground up. So in this lab data rate is one of the first steps. Another component are these Scylla devices, which we are currently developing, which also get like a semantic layer, so that they, for example, tell the system what device they are, what vendor, the manufacturer they were built from, at which date, which type of instrument it was, which measurement it can do, and things like that. So by that, we add another automated component of the semantic knowledge graph that is built up. A third layer is built up through this, what I showed to you here, these substance libraries. So whenever you add a substance to this, to this database, so here to add a substance, then an ordering process can be triggered. So you can make an order process. So you can have like a substance order, or anything, like a sequence order process, or a material order process, or an organism order process. So it's like a store book system, where you can first of all send to someone who's ordering the stuff then the information that you want to order it. Then this guy or she will pick this list, just send it to the vendors, that you get your substance, your parsed devices back, and then they are fed the information that it's ordered is going back into the database, in this later database, and you get like you have like the information when it was bought, and which price it was at, and things like that, and which order number and product number and things like that. They are then stored here in this substance instances or parse instances, and by that you build up basically your whole knowledge graph, because when you order a substance, then the name of the substance is sent to several databases, like the PubChem database, and then the system behind the scenes looks, the LARA system behind the scenes looks for information, extracts the data, and then enriches its own database with the data that comes from external databases like PubChem, Uniprot, Wikidata, and things like that. So in this workflow, working with your data, planning your experiments, you automatically generate a lot of metadata. So it's also stored who did this ordering, who did this this experiment, and things like that. So a lot of this information is then combined and added to the experiment, and then makes this whole thing searchable and structured. Okay. One word to the ontologies. So to make it really, it machine understandable, this whole database, we need to specify terms that are machine understandable, and therefore there's whole, standard available, luckily, that is called RDF, which I mentioned before. And there's also a way to describe relations between different terms in a kind of class relationship. Also, you can even specify deeper. So you have like parent-child relationships, or if these terms are belonging to the same group, or if they are disjoint. So this disjunctness. So things like that can be disjointness can be expressed with a language that is designed to describe ontologies and these relations, which is called OL. We are using that language to describe our ontologies and make these ontologies very rigid in its logical structure, or very decidable. And then you can apply logic reasoners, and you can, for example, derive. So once you make this whole tree of terms, that, for example, a protein, because it's an enzyme and because it's it has a sequence of amino acids, you can derive from this logic tree that proteins consist of amino acids because you have the right relations to that, things like that. So a lot of these different things. What we are heading at is that when you do machine learning, that the machine learning algorithms, they extract this semantic information and build their own models with it. That's the overall goal. So for building these ontologies, we're using the EMO, the European Multi-Perspective Ontology, as our top-level, mid-level ontology. More if questions are appearing, there's a Python representation of it. And we built a pipeline for the development of ontologies. Put our ontologies here on GitLab and Open Source Lab. So GitLab/Open Source Lab, so it's easy to find, and they will be maintained there. Once you have then these ontologies as these ontologies and your data represented in this triple stores, you can use the SparkQL engine, and this is here integrated into the surface of LARA. So if you go here to SparkQL query, you come then directly to the SparkQL interface and can run some queries. You see here, this is live. I mean, it's not like I'm not mocking up things. So so you have then a direct access of the SparkQL and the triple store in your interface, in your user interface, and do some queries. Storing data, linked data, is an important thing, and we were exploring what type of how to structure data and how to save data if we need to save it in a kind of file-like format. And for that, we developed, or currently develop, this SciDat format, which is a combination of tabular data. So if you have the tabular data, like a data frame data, and the metadata stored in JSON LD. So, and what you can do with SciDat, it's a Python library that combines this JSON LD metadata, the linked metadata, so the semantically annotated metadata, with the tabular data, and compresses everything into a very performant and very memory efficient file, which you can store on your drive or in file storage, like an S3 storage. So it's optimized for S3, or it's very commonly used in this Amazon-like file storage. LARA comes with one of these open source file storage. So MinIO is used to store files. And the reason why we decided to use this type of file structure is because loading CIDAT or the fast Parquet files, it's basically Parquet in the background, is immensely performant. So you can load like gigabytes in a fraction of seconds. So it's amazingly well performant, and it can combine JSON LD, so linked data with tabular data in one file basically, and that then you can efficiently store it and link it.
Sorry, Mark, to interrupt you. So the main advantage of CIDAT over NEML is the speed, or?
It has two advantages. It has or three. It has first of all, it has all this semantic information in it. So for example, if you measure like a time series, then you have like in your in this file a description of what are the tables, the table names. So what is the meaning of them, and they are referenced to ontology terms, so to external ontologies. So for example, if you have a time series versus absorbance, then in this file it's already stated that this column, the first column, which is called time, is referenced is a series of time data points, and time is defined according to this in this ontology, and the absorbance is defined according to another ontology term in pointing to this ontology. So there's a direct link between the data and its definition of the data. Of course, the definition is not inside of the file, but the reference to the definition is there. Okay. And then the speed, and it's like I mean, there's no so it's described as there's no transformation of the data when you load it into like reformatting. It's really like a memory dump from the hard drive into the memory. So it's ultra fast. Okay. And the JSON LD part of it is immediately accessible or dumpable into triple stores. So you don't need to do like an XML parsing like an animal.
Okay.
Yeah, so that's the merit. And then what you do with this data, we connect. We have a very deep interface, very far performed interface for Jupyter notebooks, for example, that you can then extract your data, query your data, and do some reporting, printing with it. We're using very how to say a stable software stack, a large software stack, unfortunately, but a very, very elaborate one. So Python, Jupyter, GitLab, Scylla, Docker, Kubernetes is then the deployment or is the planned. And for the front ends, we're currently developing Vue.js surfaces. Okay. I think with that, I stop. Let's let the rest to the questions. There's not so much more to say. Maybe to the outlook. So we are currently exploring voice control with OpenAI Whisper, so that we can get feedback from the users through OpenAI Whisper, so the text to speech as a speech to text conversion, and then hopefully we can in the long run use large language models to replace the very complex SparkQL queries, because this is something that you don't want to write as a normal biochemist or physicist or whatever. You want to have like a natural language formulation of your queries for the database, and we collaborate with people that explore right now the potentials of large language models for that. I think that's one of the most exciting parts, developments that we explore. So with that, I would like to thank a lot of people that are involved, from here, Stefan Mark, of course, and then Uwe Bornscheuer, our group leader here in Greifswald, Theo Berlin people, Peter Neubauer's group, Stefan Born, and the Kiwi team, the CLAR team, the animal team. So a lot of people are involved in that. And with that, I'll close, and if there are questions.